Hello, this is Unit 7, and we're going to be talking about growth, chromosomes, and cell division. We won't get through all of this today, but we'll get through at least growth. So let's get started. So when this eagle laid eggs to create these chicks were born, when these chicks were born, they already were made of thousands and thousands, tens of thousands of cells. And we're going to talk about how those cells were produced through a process of mitosis. So you can see when an egg is fertilized, this is the general idea of what we see, and then that one cell divides into two, those two cells divide into four, and these four cells are all genetically identical until you get to many cells, until you get to hundreds and even thousands of cells. So there's two main reasons why a cell divides rather than continuing to grow. If you've got a cell that's this size versus a cell that's this size, if you have waste in the very center of the cell, you can see how there's a much shorter distance to get out of that cell than there is from the very center of this cell. Whereas if you've got nutrients, let's see, nutrients would be good things that would go into the cell. So if you've got to get nutrients into the cell, getting to the very center portion of the cell is a much longer distance than getting into a cell that is a bit smaller. So a larger cell becomes less efficient. So the efficiency goes down trying to get the nutrients in and the waste material out across the cell membrane. As the cell gets larger, the demands on the DNA also increase. So imagine being a cell, like we said before, a large cell, and you've got your nucleus here, and you've got your chromosomes inside this nucleus, and that genetic information is responsible for all the organelles that are out in this space. They're responsible for organelles like, I don't know, like the mitochondria, or if this were a plant cell, they would be responsible for things like you'd have to have lots and lots of chloroplasts in this cell. So you can see that it becomes a whole lot more burdensome on a cell when it is larger in size. So DNA is a nucleic acid. DNA is a nucleic acid, and it's found, nucleic acids are found in the cells of all living things, all cells of living things. DNA codes for all genetic information, so all of your genetic information, all of your genes are all contained in the DNA. It codes for all the genetic information. In a typical human cell, DNA is about 2 meters long, and the way that we get it into, or get all of that DNA packed into the nucleus of a cell, is it coils and it supercoils. And we're going to talk about this structure a bit so you can see how the DNA coils back on itself. And this is not the double helix. This is coiling on top of the double helix. So if you take a piece of yarn and you coil it and you coil it and coil it, you twist and twist and twist. I think I've shown you this before, how it'll coil back on itself. And then you take those coils and coil them again. And that's exactly what happens. We start out with this structure, and this structure is called the double helix. And the double helix is made of adenine, guanine, thymine, and cytosine. And what happens is that is then supercoiled. So Like I said, in a typical human cell, the DNA is about 2 meters long. DNA is packaged into condensed chromosomes. So now, DNA in a prokaryotic organism versus in a eukaryotic organism. In a prokaryote, the DNA is circular. It's still a double helix, so we still see something like this. So we still see the double helix in a prokaryote. The DNA is free-floating in the cytoplasm. It's not in the nucleus. And they generally have one chromosome. So you can see here this prokaryote's got one chromosome and there's no nucleus. And we've got the cell membrane here. Get my picture out of the way there. Eukaryotes are, the DNA is linear. It's a long strand. So when I was talking to you about coiling up that single string of yarn, a straight string of yarn would be very much like DNA in a eukaryotic organism. DNA is enclosed in the nucleus, and generally eukaryotes have several chromosomes. They can have anywhere from 4 chromosomes, as in a fruit fly. Some fruit flies have 8 chromosomes. Humans have 46 chromosomes. This you should memorize. Humans have 46 chromosomes, or 23 pair. Bananas actually have have 33 chromosomes. So DNA wraps around histone proteins. So if you've got 4 histone proteins, what happens is DNA will actually wrap around those histone proteins, and then you get another set of histone proteins, and the DNA will wrap around those histone proteins, and so on. And it continues to wrap and wrap and wrap and wrap. And in each of these loops, there are 4 histone proteins. And what actually happens is in this one, there are 8 histone proteins and 2 wraps, 2 wraps of DNA. DNA. Let's put 2 wraps of DNA. Let's see, let's put those loops back around those histone proteins, and let's put that histone protein back as well. So we've got 4 there. All right. So DNA wraps around proteins called histones, and they condense into chromatin. The stupid association I give with that is chromatin is held in a tin can. That tin can is the nucleus, because it's loose. So you want to hold it actually in your hands, because it's just loose threads, or looks very much like loose threads, just like you see here in this area here. So chromatin can form supercoils, and those supercoils can actually, what happens is you have these coils that we talked about with histone proteins will coil up, and what will happen is they'll get tighter and tighter and tighter, and we get these supercoils, and these supercoils will go back and forth on each other, back and forth and back and forth and back and forth. And they're very, very organized, and we get something that ends up looking very much like this. And so a chromosome is one long continuous thread of DNA that consists of numerous genes. Now, there's a couple of things that I want to point out.